presentation is a study for the novel energy efficient approach for reclaiming acetonitrile in peptide manufacturing. Standard forward-looking disclaimer. Just as an intro, PolyPeptide has partnered with Donaldson for improving sustainability and aiming to implement this technology. We did put out a joint press release at the start of this week, so that is available on each of our websites, and I'd encourage you to go to each of our booths for more information as well. Just briefly, I'll take you through a background for acetonitrile generation and recycling, what some of the current issues are with that, and the proposed solution. How this impacts our green footprint as well. Just a little bit about PolyPeptide. We're an end-to-end CDMO for peptide manufacturing, manufacturing custom projects for preclinical all the way through phase 3, and then with commercial and generic peptides as well. We have a mix of production sites, some at smaller scale and some at much larger scale. I'm currently based at the Torrance site, where we do mid to large scale peptide production. And with the coming of the GLP-1 analogs, production has increased across the board. So this discussion for a greener and smaller footprint becomes more relevant than ever. As part of the green portfolio, our first aim is really to reduce usage. So wherever we can, reductions in washing, bed volumes, really reducing the overall PMI. But our next avenue is recycling. So through this technology, it really fits well within our portfolio as one of our main production solvents to aim to recycle that. Some background on acetonitrile. This is a critical solvent used in peptide purification, amongst others in oligonucleotide synthesis, where it's heavily used. Manufacturing is a byproduct of acrylonitrile oxidation reaction production. So that being a secondary product in industry can lead to some problems when other industries turn down. If you've been in the industry long enough, you've lived through a couple of acetonitrile disruptions, with a major one in 2009, as well as some minor ones in 2014, 2015 as well. The supply chain is really carbon and energy intensive. And the demand is really only growing with the growth in peptide projects such as GLP-1s. So current handling methods, if we're able to actually recycle them, they involve limited recycling and multiple distillations. It's very energy intensive, and there's some further drawbacks to that as well. Disposal methods typically involve pretty heavy treatment with multiple distillations prior to being able to either incinerate that and use some for industrial wastewater. Some of that can technically count as recycling, but I don't think burning in a cement kiln is anybody's version of green. We also have a long supply chain for a really long tail there for disposal. So that just adds to the footprint. So a typical disposal process or recycling currently involves a multi-stage distillation. Each of these has a very high energy utilization. And one of the drawbacks is using a single technique. That can heighten risk for carryover if there's any solvents with similar boiling points in the process, as well as peptides within the process that could be retained throughout that. Other volatiles can remain, and throughout this process, additives are typically required either to break azeotropes or additives for foaming control. So that requires further control of that within your raw material stream. So really having that higher environmental footprint for recycling and reuse currently, we need to find a better way to do this. So our goal is to create a sustainable, energy-efficient way to recycle acetonitrile and having robust controls within that. So Donaldson, in partnership with EcoSeed, developed a novel technique to reclaim acetonitrile. Donaldson is a worldwide expert in filtration solutions, and this is right up their alley. So they collaborated with us on installing a pilot scale trial with the goal of industrialization. And really the aim here is to benefit the entire peptide manufacturing industry. This isn't something we're looking to hold within our group. We want the whole industry to benefit with this. So what they came up with was a three-step purification process where the used chromatography solvent goes in. It's subject to filtration where we do a pH adjustment, and then a coarse filtration followed by ultrafiltration. Then that does go to a single step distillation, so we do minimize some of the energy usage there, and then to a pervaporation step, at which point you have your recovered acetonitrile. So the incoming stream, it's really important to characterize what we're bringing into the system. We need to characterize the mixture of acetonitrile, pH, any salt composition, UV characteristics as well. One of the things with chromatography is in order to separate different impurities, you likely have multiple salts or chromatography systems going in as the input for this. So we need to be able to make a design space around that and to be able to handle that process variation. So we've created a prototype system that enables us to isolate the individual stages for optimization. This really has the ability to let us create that design space around each before we go into scale up. Really important to find these things on the small scale before doing that at the ton scale. So digging a little bit deeper into each of the unit operations, the first one is filtration. We need to pH adjust the incoming stream, removal of solids in order to not foul any membranes and preserve life there, optimizing the flux, temperature, and then endpoints for water and solvent content. We really want to have the correct endpoint and feed into the next stage so that each stage is the most efficient as it can be. So for each of these, we are doing intermediate characterization, and that's important to drive the next step. So for distillation, we do have the single step of distillation in there. One of the keys here is optimizing temperature, pressure, and really what that endpoint is. We don't need to drive this within this step to the perfect endpoint, so we are able to manipulate that so it's the most energy efficient step that it can be. And then last, we have a pervaporation step. So with this membrane, we can be very efficient, optimizing flux, pressure, temperature, and then here we get the end product. So that we'll need to go for QC characterization. So the goal within all of this is having acetonitrile that meets our current specs. Really, if we can't use it within current filings, current streams, it does become of limited value. We are putting this right back into the buffering system, so we are mixing it with water right off the bat. But really, this is our typical characterization scheme for acetonitrile and release there. We are doing additional characterization for water content, as well as GCMS to identify at this stage, especially any carryover impurities that may be getting in there, and anything that we do need to optimize out of the system. So one of the byproducts of this will also be industrial wastewater. We don't want to create another waste stream while recovering the acetonitrile that all of a sudden becomes more expensive to destroy. That would be self-defeating. We have our wastewater through this tested by Pace Environmental Labs with the standards set forth by the LA County Sanitation District. So being in California, it's definitely one of the most stringent wastewater requirements around. Some other considerations that we do have to factor in would be nitrosamines, obviously doing the risk assessment and testing there. We do need to ensure that we have negative control for product carryover. Extractables and leachables for all the wetted components within the system. That can be really important as you move from different concentrations of solvents within each unit operation, especially as you get towards the higher end and extremely pure acetonitrile. PFAs have also become definitely a discussion topic and something that we'll have to characterize within wastewater treatment with upcoming regulations. So as we look towards industrialization, how do we generate value here? Each stage is going to have a certain output that we can move forward, but we also do have some waste related to each stage. What we've seen so far from the ultrafiltration is a greater than 80% recovery, so we do have some reject there, but vastly minimizing what is required for actual disposal there. So that minimizes some cost there. Through the distillation, the main byproduct from this and pervaporation is the water. So being able to put that through the industrial wastewater treatment is a real gain there and minimizes cost. At the end, we're left with the acetonitrile that we've recovered, and hopefully that's a gain. Everybody wants to go green as long as it's cheaper than the current situation. So that's the aim for all of this. So one of the things that we were looking at within sustainability was different operating models. How would we set this up on the commercial scale? We have our base case with our current generation and disposal model. That has a lot of carbon footprint from not only the raw material generation, but also the transportation and additionally the waste. There were three other models that we looked at. One was a mobile installation. This had the highest option of the three, didn't really confer too many benefits. So we moved on to looking at a centralized processing method. This could theoretically handle multiple streams from different suppliers and really achieve some economies of scale there. That does offer some benefits, but does bring in some complications with handling those different waste streams, the QC testing, and control within that. What we're leaning towards is the installation on site and treatment directly within the plant without it even leaving. So that's our best case. It minimizes, obviously, the raw material generation costs, as well as a lot of the energy and definitely with the transportation tail. So again, we really aim to minimize the supply chain here. Some of this acetonitrile can come as far as from the Gulf Coast or Ohio, so it's really trucking carbon intensive. So being able to change the dynamic with the bulk of that and only using 20% for a makeup would be a pretty good outcome from this. So in conclusion, acetonitrile remains a critical solvent for peptide purification amongst other uses. It's really a growing market, and developing a sustainable solution that has the robust quality and control is key. This multi-stage membrane technology, I think, gives key advantages over current technologies in consumption and process robustness. So we really want to have this technology out there in the industry and aim for its broad use. So, thank you. Oh, some acknowledgements for the team. So.